Roger Ngatu. I'm Associate Professor at the Department of Public Health, the University of Kagawa, Faculty of Medicine here in Japan. I would like to introduce one of my research projects about Bio Modulators of Filaggrin and TNF-alpha that we propose for the prevention and treatment of chronic inflammatory skin diseases. So I'll summarize. So this slide shows two pictures. One of the pictures is about me teaching at the School of Medicine here at Kagawa University. And the other one is from a newspaper, a local newspaper, Kochi Shimbun, Kochi newspaper, about my first discovery of the research topic about chronic skin diseases. That will be the focus of today's presentation. So in 2012, when I was graduating from a PhD course at Kochi University here in Japan, so I received first the Best Research Award, the picture is over there, from the Dean of the Faculty, the School of Medicine, Kochi University, I'm here. And the same year, I received also another research award from the President of Kochi University in Japan. So I represented my faculty, the Faculty of Medicine, the School of Medicine, and you have researchers coming from other faculties who also received the award. So the focus of my speech now will be this newspaper. It's a Congolese newspaper published in Kinshasa, La Prospérité. And this newspaper published about my 10 years long research project about the discovery of biomodulators of Filaggrin and TNF-alpha as a polyvalent treatment for chronic skin diseases. So the article is in French, because in Congo we speak French. I'm Congolese, and I will try to introduce now the biomaterials that we have discovered having anti-inflammatory properties and anti-ulcerative properties. So when we conducted the first study, the first animal experiment and the first pilot clinical trial using our first biomodulator of Filaggrin, MX. So we were interviewed by journalists from the NHK. NHK is very known in Japan. It's a TV channel. They visited our department, our laboratory, and they interviewed me. So those are the pictures taken during the TV news program. And related to the same discovery, so Kochi University magazine published an article about me, and also Kochi newspaper. And this is the National Patent of Invention delivered by Japan Patent Office in 2015. So my name is here, and my supervisor at that time when I was a PhD student. And this is the International Patent of Invention delivered in Switzerland, Geneva, by WIPO. And this is the first book published that contained most of our researches research project, experimental and clinical studies. It was published by Springer Nature in 2018. So here I'm introducing some of the biomaterials from plants, edible plants, that we are using in our research on chronic inflammatory and ulcerative skin diseases. Bio Modulators of Filaggrin and TNF-alpha, Bio MFTA. So number one is Amex, or Amex in French. It's from amygdalina, which is a plant that is commonly used in traditional medicine in some countries of Africa. And the one I'm using is from Congo DRC, in the western part of Congo DRC, Congo Central Province. And this plant is used for treating cough and other diseases. But I was the first researchers to use it for skin diseases treatment. So MX is the extract. We use both the alcoholic extract and water extract that we, in terms of improving the production of filaggrin. Filaggrin is a principal protein of the skin barrier, which protects the skin from environmental factors. So the brownish part of the skin shows through immunofluorescence technique, shows the concentration of filaggrin. So after exposure to TNCB, which is a chemical compound that induces a dermatitis on the skin of mice. So after inducing the dermatitis, we applied MX and Vernon, according to the group, or buffer water in the control group. So if you see after some hours, filaggrin is almost not there in the outer layer of the skin in the control group, but we see filaggrin concentration increased in the Vernon group. And for AMX, the lower concentration has very little expression, but the higher concentration shows an improved expression of filaggrin. This is one of the mechanisms to heal wounded or injured skin, is the improvement of filaggrin production. If filaggrin production is improved, it means the wounded skin can heal more quickly. And here we summarize the anti-inflammatory mechanism of bio-MFTA, which means Bio Modulator of Filaggrin and TNF-alpha. So we have the skin here. You have the outer layer, the stratum corneum, the middle one in the epiderma, stratum granulosum, stratum spinosum, stratum germinativum. So filaggrin is in the outer layer, and the application of topical application of MX, Vernon, and Saccharin will increase the production of filaggrin. By increasing the production of filaggrin, it will improve the function of the skin barrier. So one of our experimental studies show that even Saccharin also increases the level of messenger RNA for profilaggrin, which is the precursor of filaggrin. And even in another mouse animal experiment, so you see Saccharin here, lowering Th1 and Th2 cytokine levels, even TNF-alpha. Here, it's about TNF, IL-4, IL-6, and IL-10. So by improving, apart from improving filaggrin production, when the molecules contained in AMX and Saccharin also, when it enters through the injured skin, it will have some effect. For example, inhibiting the production of some Th1 and Th2 cytokines, thus decreasing inflammation, reducing inflammation. Particularly, they will inhibit the production of TNF-alpha. So, through this mechanism, Saccharin, AMX, Verno, and their combinations will improve the healing process and the recovery process of the skin, the injured skin or wounded skin, with the inflammation, ulcer, or skin burn. This is part of a result from our pilot clinical study. It's in French, sorry for that. But here we have the severity score of the dermatitis in human subjects suffering from dermatitis, either allergic dermatitis or irritative dermatitis, contact dermatitis. And here the severity of itch or pruritus. Most of the patients suffering from this disease, we tend to have itch and they scratch. So we have, for the severity of dermatitis, the comparison between MX1, which is water extract, MS2, alcoholic extract, compared to steroids. Steroids here represented by dexamethasone, and also we have Vaseline there. So for Vaseline, very slight change occurred in patients from the Vaseline group in terms of severity of dermatitis. But for AMX, you see a very huge difference compared to Vaseline. And AMX and dexamethasone had similar efficacy in the treatment of dermatitis, especially AMX2, the alcoholic extract, had almost the same effect with dexamethasone. But regarding itch score, severity of itch, AMX 1 and 2 had very good anti-itch effect compared to the steroid and Vaseline. Our skin could appear quickly. This is one of the index cases that we had during our pilot clinical trial, the first one. It's an albino guy, an adolescent albino in Africa who has been suffering from this kind of skin disease that we call nummular eczema. So it's a contact dermatitis, but albino people develop due to exposure to sun rays. So it's a photodermatitis. So he was taken care of at health centers using a steroid preparation for a very long time, but there was no improvement. So during our mobile clinic activity in rural Congo, so I myself took care of this patient. So I used MX. So we applied MX every day, and 3 weeks later, everything was okay. So let me introduce now two cases of chronic inflammatory skin disease, especially ulcerative skin disease. So this is a diabetic patient, a female diabetic patient, 33 years, in Congo, who has been suffering from diabetes for a long time, and also has one of the complications. This ulcer was treated, but there was no improvement for over 1 year of treatment using antibiotics and wound care with antiseptic solutions. So we treated this patient with AMX. With AMX, this was the second day of AMX treatment. On the fifth day, so there was a little improvement, and 1 week later, day 10, more improvement, and 2 weeks later, almost disappeared. The wound almost disappeared. And this was a very severe case of diabetic patient, about 52 years old, Congolese male diabetic patient, who had this ulcerative complication for more than 1 year and a half. It was on the foot, and he underwent 2 amputations. There is no 2 here. It was amputated because of the complications, infectious complications, etc. And there was no good evolution of these lesions. So when I was contacted, because I know this patient, and he knows me, one of my colleagues when I was school student, so I provided the second product that we use, AMX and LIM. And for 3 months, we took care of him from the deep wound that we had. Sorry, the picture are not very clear, taken by the patient himself. A large and deep wound, so you have almost recovery of the skin, the wounded skin, with re-epithelialization of the affected area after 3 months of treatment. So that's all for today. So this presentation was focusing on the biomodulators of Filaggrin and TNF-alpha that we are using in our research for